I'll talk to you guys briefly about what I've been working on. Pretty much TB treatment response. And this has been my PhD project so far. Pulmonary TB is tuberculosis with lung inflammation, and Mycobacterium tuberculosis is the pathogen that causes TB. It infects the lungs. As such, the lungs is the ideal tissue for studying transcriptional changes in TB. What's the problem in this project is that lung tissue samples are not readily available and difficult to extract, but blood is easily accessible and readily available. So our aim for this project was to identify biological processes underlying the resolution of lung inflammation during TB treatment. So we set out to constrain peripheral blood transcriptional changes to correlate with PET-CT FDG uptake in the lungs while correcting for cell type abundance. I'll explain some of those terms in a bit. What's PET-CT? So PET-CT is a medical imaging instrument. It's used to measure metabolic activities in the body. And it measures FDG uptake. FDG uptake indicates glucose activity in the body and hence is used as a surrogate for inflammation. So we performed RNA-seq and 2 PET-CT measurements on TB patients that were cured at the end of treatment. So we performed RNA-seq and collected PET-CT measurement during treatment at those time points at diagnosis, month 1, and end of treatment. So to constrain RNA-seq gene expression data to correlate with PET-CT measurement. Why? Because we're trying to use peripheral blood to identify biological processes in the lungs. Like I just said, to extract lung samples during TB treatment, it's not feasible. To collect lung samples at each of those time points is not a feasible task, but you can easily collect blood from patients. So hence we're using peripheral blood. So to constrain RNA-seq gene expression data to correlate with FDG uptake, PET-CT FDG uptake, we use linear mixed model to correlate transcriptional expression levels with PET-CT uptake in the lungs. Then we also corrected for the proportion of cell types from gene expression data using computational deconvolution. And we performed gene enrichment analysis on the genes that were correlated with FDG uptake over time. We identified 639 genes whose transcript expression levels were strongly correlated with FDG uptake in the lungs during TB treatment. Among those genes were upregulated genes. When I mean upregulated, I mean genes that their expression level increased over time during treatment, and genes that their expression levels decreased or their transcript level decreased during treatment. The upregulated genes were enriched in B cells, B cell activation, and antigen processing and presentation, while the downregulated genes were enriched in platelet, blood coagulation, dendritic cells, and platelet activation, as well as type 1 interferon response. Due to the size of the enrichment table, I couldn't show those tables. I've been struggling to fit those tables in this slide, but they couldn't enter. Take-home message for this, it was really interesting to see that constraining peripheral blood transcriptional changes with a measure of lung inflammation does identify biological processes that are related to inflammation. It was really interesting to know that. And these expression profiles of these correlated genes could serve as markers for monitoring TB treatment response from a clinical perspective. At the end of the day, we say that the resolution of lung inflammation correlates with increase in the expression of genes that are enriched in B cells. So we are seeing B cells upregulated during treatment, and platelet and blood coagulation being suppressed during TB treatment. So it was a really interesting finding, and we hope to see what the reviewers would think about it as we plan to publish this. I want to thank my funders, to thank Stellenbosch University and the Immunology Research Group at Stellenbosch University for the opportunity to perform this research, and as well my supervisors and the South African Tuberculosis Bioinformatics Initiative. Thank you very much.
Great. Thank you, Trust, for your presentation. An interesting one, certainly. Some very interesting results to unpack there. We've got some questions for you in the comment box. So Issa is asking, could you tell us more about the models that you use to measure the expression levels of expression? And if you have any plots or tables to share, that's okay to share too, if you want to share your screen again and show us those. And of course, thank you for the talk. So go ahead, Trust.
Could you repeat that question?
Sure.
I don't understand. Models for measuring expression level.
Yeah. So was there any particular software or model type that you used to measure the differences in expression level?
Okay, so we're not doing differential gene expression here.
Mm-hmm.
We're using linear mixed model, which is basically a linear regression model, but it performs hierarchical modeling. What it does is it takes your gene expression levels in continuous form and correlates them. So it accounts for the intra-subject correlation, which is inherent in repeated measurements. It does not behave like a typical ANOVA, which ignores and assumes a lot of stuff when you're doing repeated measurements. So it constrains your gene expression profiles to be correlated, to be consistent with your PET-CT measurement. And in this study, the PET-CT data that we had showed that FDG uptake, which is a surrogate for lung inflammation, was decreasing during treatment. So if lung inflammation is decreasing, we want gene expression to correlate with that. So that's what the model does. As it's doing that, it accounts for the intra-subject correlation. So if you take one subject over time, you have variability over time in one subject during treatment, and you also have variability between subjects. Different patients respond differently to treatment. So we want to account for all those biological features, and the model is able to account for that. So that's the main model used in this study. With respect to the model for gene expression, there was no model for gene expression. We just perform pre-processing of RNA-seq gene expression data. I don't know if you're familiar with analysis of RNA-seq gene expression data. Pretty much you do your filtering and all that stuff. So there was no model.
I'm going to stop your explanation there. For the sake of time, we will move on, but thank you. And I think if you're welcome to have more in-depth discussions during the social or just after the social if you want to get in touch. But just for the sake of time, we will need to move on. Thank you very much for your talk, Trust.